In cells, NRF2 is essential for boosting caloriculin to trigger phagocytosis, but KEAP1 binds and blocks NRF2, stopping the process. This suppression halts caloriculin expression. Then, enter the predator. Moringa activates NRF2 release. Now NRF2 is free to boost caloriculin, immune system.
In this video, I'm going to be talking about KEAP1, KEAP1, as well as NRF2, and how this could actually have a possible effect on CD14 and toll-like receptor 4. So TLR4 is basically what's activated by lipopolysaccharides from gluten ingestion. Also, CD14 is something that is on the toll-like receptor 4, and it can either be cleaved off or it stays on the toll-like receptor 4. And there are different effects as to if the CD14 is cleaved off or if it's intact on the TLR4. So there are different effects when NRF2 is activated, and one of them is something called TRXR1-6, or short for Peroxiredoxins, and these are cysteine-based peroxidases. So they actually these protect and shield CD14 and toll-like receptor 4, as well as KEAP1 from oxidative damage. So these peroxiredoxins, they detoxify peroxides such as H2O2 and lipid hydroperoxides before these oxidants can attack the protein thiols on the CD14 and toll-like receptor 4. Now, there's something called Thioredoxin, or TRXR for short, and this protects the CD14 on the toll-like receptor 4 from oxidative cleavage. This ensures proper TLR4 endocytosis. Now, endocytosis is another word for immune regulation and proper immune function. Okay, if excessive peroxides are not detoxified, this can actually inactivate TRXR, therefore leading to immune dysfunction. The peroxiredoxins, and there is PRX1 up to PRX6, those are supposed to detoxify the peroxides, such as hydrogen peroxide, lipid hydroperoxides, and then another one called peroxynitrite. The lack of detoxifying peroxides can also lead to hyperinflammation. And a cancer is known to, or cancer cells are known to gravitate towards locations of inflammation. So it's important and it's critical for the peroxides to be detoxified. So if peroxynitrites are not detoxified, that will allow NRF2 to escape degradation, because KEAP1 holds NRF2 hostage. And if the peroxynitrites are ongoing and excessive, that could lead to NRF2 promoting oncogenes and even tumor growth. So it's no wonder that KEAP1 is holding an NRF2 hostage, because KEAP1 or KEAP1 is actually a sort of a way to protect against cancer, because KEAP1 is trying to prevent NRF2 from being overactive, so that the NRF2 does not upregulate oncogenes and leading to tumor growth. So KEAP1 can also lead to NRF2 being degraded if KEAP1 holds NRF2 long enough. So the peroxynitrites, um, they can also cause DNA damage, which is cancerous, and also mitochondrial dysfunction. So that also means that your immune system will also be dysfunctional because the immune system needs like a ton load, a huge amount of ATP. And if the mitochondria is dysfunctional because of peroxynitrites, then there will be no or not enough ATP for the immune system. Okay, so also because of peroxynitrite, persistent NRF2 activation, the KEAP1 isn't holding it hostage long enough, is that it promotes the survival of damaged cells, confers chemotherapy resistance. So if NRF2 is overactive, which is also why the cancer patient had oncogenes or tumors to begin with, the chemotherapy will not work or will be resistant if the NRF2 is activated. But the reason that the person had cancer to begin with, so that the chemotherapy would be resistant, is because of the tumor growth. So the chemotherapy was never really like a benefit or was not necessary to begin with. So another issue with excessive uh, NRF2 is that it dampens necessary reactive oxygen species signals that are required for immune signaling. So, excess NRF2 weakens the immune system. So, it's no wonder that KEAP1 is holding NRF2 hostage. Also, the apoptosis in cancer cells also requires reactive oxygen species signals. Also, excess NRF2 hyperactivation as a result of KEAP1 being degraded. KEAP1 can be degraded because of peroxides not being detoxified. So, NRF2 overactivation also leads to M2 phenotype macrophages. So, those are the macrophages that actually support and protect tumors. Okay, so this is why KEAP1 has to hold NRF2 hostage at times, but not all the time, to prevent the oncogenes. But KEAP1 will completely, totally lose the ability to hold NRF2 hostage, which is not good, if the peroxynitrites and also H2O2 and lipid peroxides are not detoxified. Because if they are not detoxified, then they will eventually degrade uh, KEAP1, and KEAP1 will lose its ability to degrade NRF2, and then NRF2 will escape degradation, therefore oncogenes and tumor growth. Okay, now I'm going to be talking about the effects of Moringa powder. So it has uh, quercetin, vitamin C. So Moringa, the direct antioxidant action is that it scavenges H2O2 and lipid peroxides, therefore preserving the protein thiols in the KEAP1 and even in the CD14 and toll-like receptor 4. So this means that the uh, the lipopolysaccharides can bind onto the toll-like receptor 4, but that's a different topic for a different video because if the peroxides are not detoxified, then that leads to impaired LPS binding as well as internalization of the toll-like receptor 4 and the CD14. So Moringa will prevent the hyperinflammation of the toll-like receptor 4 caused by the lipopolysaccharides, which is a gram-negative bacteria. Also, the Moringa will help the KEAP1. So Moringa allows KEAP1 cysteines to to control the activation of NRF2, but without KEAP1 being totally degraded, because of the peroxynitrites not being detoxified by Moringa if there wasn't Moringa around, because then the oncogenes would be activated, which are cancer genes, if KEAP1 cannot keep NRF2 under control. So that's it for the video. Like, share, comment if you want more future videos, and I'll see you in the next video.